PCR creates new DNA fragments in a cycle of 3 steps. The first step, denaturation, involves heating the solution in the plastic tube to a temperature just short of boiling, so the individual DNA strands of the template strand separate or denature as a result of the breaking of hydrogen bonds between the complementary bases. The second step, annealing, begins as the solution is cooled. Because of the great excess of primer molecules, the 2 primers bind or anneal to their complementary sequence on the DNA template. In the final step, extension, the solution is heated to the optimal temperature for DNA polymerase, and each primer is elongated or extended by means of the deoxynucleoside triphosphates. After sufficient time to allow new DNA synthesis, the solution is heated again, and the cycle of denaturation, annealing, and extension is repeated over and over, usually for 25 to 35 cycles. In each PCR cycle, the number of copies of the targeted fragment double. The first round of PCR amplifies the targeted region into 2 copies, the next into 4, the next into 8, then 16, 32, 64, 128, 256, 512, 1024, and so forth. The doubling in the number of amplified fragments in each cycle justifies the term chain reaction. Although PCR is elegant in its simplicity, the DNA polymerase enzymes for many species, including humans, irreversibly lose both structure and function at the high temperature required to separate the DNA strands. At each cycle, you would have to open the tube and add fresh DNA polymerase. This is possible, and in fact, this is how PCR was done when the technique was first developed, but the procedure is time-consuming and tedious. To solve this problem, we now use DNA polymerase from a bacterial species, THERMUS aquaticus, that lives at the near boiling point of water in natural hot springs such as those at Yellowstone National Park. This polymerase, called Taq polymerase, remains active at high temperatures. Once the reaction mixtures are set up, the entire procedure is carried out in a fully automated machine. The time of each cycle, temperatures, number of cycles, and other variables can all be programmed. The fact that DNA polymerase from a bacterium that lives in hot springs can be used to amplify DNA from any organism is further evidence for the conserved function and evolution early in the history of life of this enzyme.